Welcome to this lecture course in optical microscopy. So this subject is one that's close to my heart. I do research. My group, the Laser Analytics Group, does research on developing new technologies in optical microscopy. And you may ask yourself, why a course in optical microscopy? Well, first of all, it's a fascinating subject. I find it incredibly interesting. In many ways, the history of the microscope mimics the history of science itself. Many of the greatest discoveries are made using the optical microscope, and to this day, Nobel Prizes are won in the field of optical microscopy. The other point is that in this department, in chemical engineering and biotechnology, we do a whole range of different research activities, and microscopy is likely to be important for almost any of the research activities that are ongoing in this department. So I hope in this slightly research-oriented course that you understand the principles, find it fascinating, and learn about concepts that may be useful also in other contexts. And so I begin with the first slide to just give you an overview of what the course is about. So in this first lecture, and maybe the next, I will just give you a brief overview of different concepts. I call this a short history of the light microscope. And in doing so, I will introduce the important concepts that we need to deal with. And these are diffraction, resolution, and contrast. And defining those in the beginning will help us then to develop instrumentation and technology and understand it in such a way that we can deploy the methods in the best possible way. Then I'll give you an overview of the basic elements of a microscope and then begin with the theoretical concepts, which will take up quite a big part of the course, maybe the initial half, before we go into applications and advanced techniques. There are a number of great websites you can look at to look at concepts in detail. So the first one named here, the Microscopy Primer website, is absolutely outstanding. It has lots of interactive tutorials on there which make you sit in front of microscopes almost. It feels like that. So you can test different concepts in realistic conditions. And most of the things that we talk about will be in some way explained also there. So I highly recommend you looking at these sites. There are many links here, but because different manufacturers have slightly different variants, but essentially these are similar contents on these. There are many books on the topic, but either the books are too general or they are too difficult. And so there's really not a book that you really need to consult in this course, but I list these books down here in case you will do a research project that involves microscopy, and then you can consult these books for further and more in-depth information. If you want a bit of a primer on optics in general, especially on the method of fluorescence, I recommend a chapter in this book here, Grant Fowles. There's a really good chapter in there on fluorescence and also a good chapter on the general idea of diffraction, because diffraction is super important. Optical diffraction is important in this course. Lakowitz is a book. It's a huge book. It's called Principles of Fluorescence Spectroscopy. In our field, it's simply called the Bible. If you need in-depth information about how to label sample cells and so on with different colors and fluorophores and use different techniques on them, then that's the book to consult, but it's a research book. The other two books, they are really too advanced, but they are for use in case you conduct a research project. So with that in mind, I hope you will enjoy this course. If you have any questions, please do get in touch. With these new tools that we have now, there are many ways of doing this. And I will try and respond to this also in some live sessions that we will have together, where we can do Q&As and go through some concepts that you may have questions on. So without further ado, I will begin by giving you the first part of this course, a short history of light microscopy.